Thank you, Aidan, for that nice introduction. Thanks to you and Matt and to the Institute for inviting me down. I've been having some great talks here and really enjoying the visit. So I just wanted to comment first that I think great universities all have birds as their mascots. And so wonderful to see the Oregon Ducks here. Where I'm from at UBC, we've got the UBC Thunderbirds here. It was wonderful to drive down yesterday and see all the volcanoes on the way, and so I'm having a lot of fun here. Now, usually when I present my work, or in the past, I've usually framed it in terms of speciation, so how one species becomes two over time. And I've studied groups like these greenish warblers in Asia and these Pacific and winter wrens in North America, largely asking like, how does one species become two? And what do they think of each other? Do they hybridize? Things like that. But what I've been trying to do lately is kind of frame the work more, maybe for an Institute of Ecology and Evolution, you know, this ecological question of how or what determines whether two species can live in the same area. So I'm asking, you know, in all of our work that we're familiar with as biologists and in your own systems, if you think about them, probably a dominant pattern that you see is like there's species A here and there's species B here, and maybe they come into contact somewhere, but they usually don't overlap each other's range until many, many millions of years have evolved between them. Then you can tend to get sympatry. So the question is why? Why is that? And that's what I want to focus on today. So, of course, this is seen not just in birds, but also in Mimulus that many of you study. This is just taken off Matt's website. And we see this pattern of different forms of Mimulus having very strong like geographic structure. So each place you go, you tend to find one type on this map, and then you go a little ways away to find another type. So we're kind of asking, why is that? Now, in birds, dispersal distances tend to be a lot higher than in plants. And so the scale of variation, you know, going from Mimulus, where you have all this structure right across California, to the scale of variation in birds is usually much bigger. Like we have to go over much broader spaces to kind of find this much geographic structure. You know, when I work here in British Columbia is right down there near, or Vancouver's near that little Vancouver Island down there. But a number of years ago, I decided to go up to Northeast BC to this town of Tumbler Ridge and study bird populations there. The reason is because in that region, you often see the eastern birds and western birds coming together in that narrow region. And so in that area, here's a photo of that area, we find all these western species. If you're a birder, you might recognize these locally because these species are also found in Oregon, at least in the winter or on migration. But in this region of northeastern British Columbia, you also have these eastern species that you could see in New York or Quebec or Ontario. And so why in this little region of northeastern BC do we have both sets of species? But if you go a little further east, you don't have the western ones. A little further west, you don't have the eastern ones. So why is this? Well, part of the reason is because there were ice ages. So here we have North America these days, nice green forest across most of Canada up there. But of course, 18,000 years ago, Canada would not have been a very nice place to live. Lots of ice over most of the country. And so those forest living birds that we have there now had to come from somewhere else. And where they came from were these refugia in different parts of what's now the United States. The ice melted and the birds and the forests moved north. So what you had is these two expansions moving northward into what I call the Great Canadian Suture Zone, which is a place where these two different groups that evolved in ancestry, evolved in allopatry during these 80,000 years or so of ice ages and came together. And we have a lot of people in my group have studied basically this kind of thing, these Eastern meets Western groups. And so a lot of the stuff I'll talk with you today is sort of a combined work of all these different people. So I just wanted to show them. And these are my 4 current PhD students. So just wanted to give a shout out to them. Okay, so this is the big question. For many pairs of species, there's no current geographic barrier. So we had those ice ages in the past, but that was long gone. You know, we're talking 12,000 years ago, the ice mostly melted. So what's keeping these overlap areas narrow since then? And this is just to show you there are a bunch of different bird groups. There's an Asian, a group in Asia. Here's some North American systems. Weir and Schluter did a nice study where they showed there's a typical kind of characteristic pattern in North America of a West Coast form, an interior West form, and an Eastern form. And we see this repeatedly in different groups. But you'll see a lot of them, the forms come together, but don't really overlap very much. So why is this? Well, the main explanations people have given for lack of sympatry are 3. So the forms could be ecologically differentiated, and there could be a habitat transition. So species A is adapted to a habitat here, species B adapted to a different habitat here, and they don't co-occur for that reason. Another explanation ecologically is almost the exact inverse of that, is that they're so ecologically similar that they can't co-occur in the same area because they're too similar. And so niche exclusion basically means only one can live in the area. Those explanations both fall under this term coexistence theory, and there have been some really nice papers, Chesson 2000 and others have looked at these kinds of dynamics. But there's another body of literature that kind of comes from the evolutionary biology side of things that says, well, these could be areas with hybrid zones. And if hybrids have low fitness, that could cause a transition zone between the forms. And basically, they don't expand into each other because of this hybridization. And that falls under this area called Klein theory. Now, these 2 things assume some different underlying assumptions. So coexistence theory usually assumes complete reproductive isolation. So the work by Chesson and others, I've looked for the words hybridization or introgression in those papers, and they're basically not there. So the assumption is full reproductive isolation in much of the work behind coexistence theory. Klein theory makes kind of the exact opposite assumption, that there's completely random mating between the forms. So you can see there's a big gulf between these. Completely random mating, complete reproductive isolation. So what I want to propose today is that a lot of these cases of narrow overlap are because hybridization is limiting coexistence. And this isn't a new thing. I'm not claiming to be the first to have claimed this. There are lots of people who have touched on this, but I want to talk about, I think this is a broader explanation that explains more things than maybe we were aware of. So hybridization is much more common than previously thought. There have been a lot of papers looking at the amount of hybridization in different groups. One of the results of all the DNA analysis that's been happening lately is that we can detect hybridization more, and we're finding it much more commonly than was previously thought. Second, today, I'll show you that simulations show a bit of hybridization limits coexistence. And then the third part of today's talk, I'll talk about empirical cases showing hybridization is rare in some cases, in some specific cases, but it has a very large influence. So first, just on the idea that hybridization is more common than we previously thought. When I started this line of work in British Columbia in 2004, the bird books all said these 2 are separate species, and they didn't say anything about hybridization between them. But I noticed there were these 2 ranges with only a bit of overlap on the map. And I went to that area and asked local birders, do you see these two? And they said, yeah, we see them, but I'm often not sure what they are because they look kind of intermediate and they sound like I can't predict what they're gonna look like based on what they sound like. So I, of course, got thinking and thought, oh, maybe there's a hybrid zone. And we collected data. This is just showing location from west to east. And the vertical axis is just the number of alleles they have of the eastern species. And sure enough, there is a hybrid zone right in the middle. It's about 80 kilometers wide. Easy for biologists to miss because that's not a very populated area. But I'm convinced this is keeping the two species from truly coexisting. It's just that they hybridize. Here's another case of two warblers. Same exact story as I just said. The bird books show them. They've always been called two species. No mention of hybridization in the literature, but you go to that area and sure enough, there are a bunch of hybrids in a narrow hybrid zone. Now, biologists have developed all kinds of theory and math to explain what's happening in these situations. So if you have two species and they come together, there can be a contact zone where they hybridize with each other. And if so, what can happen is if the hybrids have lower fitness than the parental forms, then you can get this demographic sink in the middle of the hybrid zone where not as many offspring are being produced there compared to outside the zone. That means there's net gene flow into the zone. And so you have low fitness, but net gene flow in, and it becomes a very stable situation. And so this is often called a tension zone because it's this balance between the low fitness causing low reproduction, but net flow into the zone. And it's quite stable over long periods of time. And the system just stays the same. So I developed some simulations to actually look at this. And I'm just gonna, so we have location along this axis. The green dots are individual birds and their genotypes are shown on the vertical axis. So how are they this species or that species up there? In the middle, you have hybrids. If we run this forward, each individual is moving a short distance. It's choosing a mate nearby, just random mating in this model. And over many, many generations, you're just seeing the hybrid zone sort of bounce around, but stay roughly a similar length. So, and that happens if the hybrids just have, 10% reduced fitness compared to the parental forms. So, Nick Barton and others have developed Klein theory, this mathematical theory, to relate the width of the Klein, so the width of that hybrid zone, to the dispersal distance over the strength of selection against heterozygotes. So essentially, as individual dispersal distance goes up, the Klein width goes up. As the strength of selection against homozygotes goes up, the Klein width goes down. And the key result here is that the lower hybrid fitness, the narrower the zone. But this math assumes, as I noted before, no assortative mating. It's assuming completely random mating. And, this was done because it makes the math more tractable. But we know in many cases that hybrids can be pretty rare in these areas. And that implies that there is some assortative mating, that individuals are reluctant to mate with the other species. And so how can we incorporate some assortative mating into this framework? Well, Dolf Schluter and I decided to tackle this problem. This was a second paper of this hybrid zone with assortative mating model that I developed in an earlier paper. And we asked, how might a small amount of interbreeding affect the ability of two species to live in the same place? And so it's this hybrid zone with assortative mating sympatric model, or HZAM-SIM. And in this model, we developed an agent-based simulation of diploid sexual individuals. written initially in R and then in the programming language Julia, which accelerated it about 60 times, 60 times. So obviously I like that. The starting point is that two populations have just come into full sympatry. Individuals have some different genetic loci, each with two alleles. There's a 0 locus or 1 locus, and they start in the two species with one species having all 0 loci, 0 alleles, the other species having all 1 alleles. And these additively contribute to a trait T that ranges from 0 to 1. Okay, I said that. And there's non-overlapping generations with cycles of mating, reproduction, and survival to adulthood. And so we can incorporate these three kinds of non-neutral things, sort of functional differences between the genotypes. We can incorporate assortative mating, ecological differences, and low hybrid fitness as we want into the model and run it in different ways. First, assortative mating is modeled like this. Each female, and it doesn't matter which sex is done like this, but I'm just explaining it with female and male playing these roles, but each female encounters a random male and can accept or reject. If she rejects, she then encounters another random male and can accept or reject and keep going on like that. The probability of acceptance depends on the difference between the female and mating and the male trait. So if there's 0 difference between the female and male in their trait, then she always accepts that male. If they're completely different, so initially the two species are different by one whole unit of difference, then the probability of the female choosing that male is represented by this H here. And we express the strength of assortative mating as 1 over H. And so if SAM equals 10, that means there's a 10 times attraction for the own species compared to the other species. That intuitively sounds like pretty strong assortative mating, right? 10x. We also have reproduction that's influenced by ecology. So there's limited resources on the environment, and each female's expected number of children is based on the competition for resources in the environment. So we use a random draw from a Poisson distribution to determine the expected number, to determine the number of children for each female. And this is the mean of that Poisson is determined by ecological competition. So there's two resources, alpha and beta, each with equal carrying capacity. And the two species differ in their ability to use the resources. So the species with all 0 alleles can use resource alpha, and the species with all 1 alleles can use resource beta. Hybrids can use each resource half as well. So there's a linear decline with this kind of ancestry index here represented by the trait. This means the total competitive ability of any individual is 1. And so there's no predefined advantage or disadvantage to being a hybrid when it's encoded this way. That's the key thing here. So all the results I show you today will be with maximal ecological difference. So this is where the two sort of pure forms of the species eat completely different things. Okay, so we're saying ecology is maximally different. Now we can also have low hybrid fitness, and this is specified as either a model that's based on heterozygosity or based on epistasis. But essentially, we have a fitness of 1. This is the probability of surviving from young to adulthood is 1. They all survive if they're of those starting genotypes. But if there's some intermediate genotype, they have lower probability of survival. We represent that by W hyb over there. Okay, so let's get to the results. What do these simulations show? So we start with 500 individuals, run for 1,000 generations, and look at the outcomes. And I'm going to show you results on a graph like this, where we have strength of assortative mating going from no assortative mating, that's random mating, 1x, all the way up to 300x, and then 1,000x, and then complete. That's, they never choose the other species as a mate. And then vertically, we have hybrid fitness with 1, meaning no cost to being a hybrid. But as we go down to 0, that's the hybrids have 0 fitness. So let's look at some different outcomes. So here, where we have no assortative mating and no lower hybrid fitness, what happens is in generation 1, these darkness represents how many individuals, the density of individuals of that hybrid index at that generation time. So here's our starting point, 2 species. Well, there's random mating in this model. So in the first generation, you get a whole bunch of hybrids. And then you get back crossing and you get the hybrids mating and the species blend together. And you see here's N up here, the number of individuals staying roughly constant over time. And so that outcome we're going to call blending. So this is just the 2 species merge into 1, right? So let's add, well now let's say there's perfect assortative mating. So this is where they never mate with the other. And we see over time, obviously, there's 2 species stable over time. Only true if the ecology is different. If they're similar ecologically, then eventually one happens to go extinct just stochastically over time. But here, the 2 resources are maintaining the 2 species. We're going to color this in with this yellow color. Whenever we have ecological differences and complete assortative mating, that's the outcome. Now, let's see if we have pretty strong assortative mating, SAM equals 10, so 10x preference for their own type. Well, surprisingly, maybe, it doesn't take long for them all to merge into kind of a hybrid group, a hybrid species. This is because despite the strong assortative mating, hybrids are being produced, you know, in maybe a fraction of the time. And they're able to survive, they're able to reproduce, mate with each other, and so forth. So eventually you lose the 2 species and only one exists. One combined species exists. So we'll color that in with that salmon blending color again. Now let's add some low hybrid fitness. So it's similar as before, except now these hybrids are produced, but they have low fitness compared to these forms. And it's like an adaptive valley then, and there's selection towards the extremes. So the population declines over time, but then evolves back towards one of the extremes. And you then have one of the starting forms kind of reemerge, but as the only form there. This was really interesting to me when we saw it, because you're losing half, like there's now an empty niche, and the population is now at half its population size compared to the start. So we're going to color this in with this, I don't know, purple color, extinction of one species, leaving an empty niche. So now let's reduce assortative mating just a tiny bit. And what happens here might be surprising. Here we have this production of hybrids, but they're low fitness, and the assortative mating doesn't allow a recovery of one of the species or the others, because it's not strong enough. And so you get extinction of both populations. Now, what we're sort of envisioning here in this sympatric model is that this kind of represents what might happen at the center of a spatial hybrid zone, is you might have an area where really the species just can't coexist at all. And so we'll color that extinction one in black there. And we can run each parameter combination many times, or 5 times, and graph the most common. And this is what we get. So that yellow area is where you have 2 species being maintained over time. And it requires like 300 times or greater assortative mating strength. So this is really strong assortative mating required to get two species. Otherwise, low hybrid fitness leads to extinction of one or both species. So that's kind of the overall outcome. And one thing that's interesting is if the results kind of depend on the intrinsic growth rate. So something like insects that have maybe lay many eggs, or maybe some plants that can have many, many offspring, there might be more region of the parameter space where you get coexistence of two species compared to something like birds maybe that only have 4 eggs per nest that have lower intrinsic growth rate. So that's just an interesting detail. But in all cases, it requires quite strong assortative mating. So the conclusions is that coexistence requires just a little bit of ecological differentiation, but extremely strong assortative mating. So Dolph and I postulate that the coexistence of closely related species is more often limited by insufficient assortative mating than by insufficient ecological differentiation. And we appear to have some good examples of rare hybridization limiting range overlap. So I'm going to talk about these 3 examples now. So the Pacific and winter wrens, actually before 2010, they were called a single species. They were just the winter wren. But in earlier studies had shown song differences between the western form and the eastern form, and also some genetic differences. So we decided to investigate that, went to the overlap area, and we found really strong song differences between west and east, represented right up here. So the songs were just as different here as they were between Oregon and New York. And also the mitochondrial DNA, extremely different. So this is a haplotype network representing mutations, 65 mutations between the species, but within each one, only 1 to 2. So this was enough evidence to elevate these 2 subspecies to species, and that's what the authorities did in around 2010. So now they're called 2 species. But run the clock forward, we collected more samples and did genomic analysis. So this is Elsa Mikkelsen over on the left. Did genotyping by sequencing of a bunch of samples. This is a principle, well, first I'll say this is a admixture plot that I think some of you have used. Each individual is shown as a column, and the inferred ancestry, blue or red, is shown for each individual. Now, most individuals have clearly either Eastern ancestry or Western, but there's 2 individuals right there and there that are perfect F1 hybrids. And you see it in the PCA plot on top. PC1 is the dominant axis. PC2 explains only a tiny bit of variation. And you see 2 F1 hybrids. But there's no F2s or back crosses or any, when we look at genotypes across the genome, we detect no evidence of recent hybridization, I mean recent back crossing. So this means we have pre-mating isolation is very incomplete. We don't have an immense sample size where they meet up there. And so to detect 2 F1s means it's not super rare. It's just that they don't have any reproduction themselves. So the post-mating isolation is complete, despite pre-mating isolation being very incomplete. And if we think about that, that could really limit the expansion into each other's range, because they're producing these 0 fitness hybrids where they co-occur. And that's got to be a cost on the population. And that cost is borne and more by the rare species in an environment compared to the common one. So we think this is really limiting the expansion of the 2 species into each other's range. So now I want to talk about this greenish warbler ring species that I started studying long ago in the mid-90s. But even earlier than that, Ernst Mayr and Claude Teisers did, Claude Teisers did work on it. Ernst Mayr wrote about it in his book and described it as this ring of populations sort of surrounding the Tibetan plateau. And these birds live in the trees. And so they're not able to inhabit the high elevation desert of Tibet or the lower elevation deserts further north of there. But there's this ring of trees around there. And so Teisers noticed that up in Siberia, the V and the P represent two different types that have different wing bar patterns. And he noticed no hybrids or no intermediate forms in that sort of overlap area near the Yenisei River up there. But he described gradual variation along the whole chain of populations encircling the Tibetan plateau. So I started investigating this for my PhD, and together with Trevor Price and a number of other collaborators, we gathered data from all the sites with little letters next to them. And there's this hypothesis that Claude Teisers put forward that they expanded along these two pathways, gradually changing as they went, and became different species up there such that they're reproductively isolated. So this is called a ring species, where you have one species boundary in a ring of populations. And so this scenario is supported by songs and calls, which we've analyzed, and also plumage. What about genes? So we've done a bunch of genetic work over the years. In 2016, we published a paper with this principal components analysis in it. So if you're not used to these PCAs, each symbol represents an individual, and the closer two individuals are together, the more similar their DNA is. The further apart, the more different they are. And so on this plot, we've got this tight cluster of West Siberian birds from that whole West Siberia to East Europe area. We've got Central Asia. We've got the Western Himalayas, the Central Himalayas, Nepal, China, and East Siberia. So that you can see that there's kind of this progression around the ring in their genetic signatures. This is based on 134 individuals, 580,000 SNPs. But we wondered, so from these data, Viridanis, the Western Siberian subspecies, and Plumbitarsis, the Eastern Siberian one, appear reproductively isolated, right? These very vastly different genetic clusters, huge gap in between them. So we wondered, if these are two species that are reproductively isolated, why are they not expanding into each other's range in Siberia? They seem to live in similar forests. What's really limiting that? So a little clue, look where the Plumbitarsis cluster is pointing. We noticed this in 2016, that it's sort of pointing that way. And we had some evidence then that there were bits of West Siberian DNA in the East Siberian birds. And so since then, we've collected more data, or I should say, not me. Nikolai Kovilov and Irina Morova collected a bunch more samples from the north. We've got more samples from this region and from a region near Beijing from other collaborators. Today, I'll focus on data from the north. And here is the new data. So about double the number of individuals and about double the number of single nucleotide polymorphisms, because we also have a new reference genome that we sequenced, a really high quality chromosome length reference genome. So we have more SNPs because the mapping is better to the genome. And you see here, so far, I've circled populations, nothing too surprising, but there's one individual I haven't circled yet, and that's this one. And it turns out this is a hybrid in Siberia, but we can look, we've looked at its detailed genotypes and can tell it's not an F1 hybrid. Rather, it's a back cross of an F1 and an East Siberian bird. So this one sample is pretty amazing because it tells us not just that there's a backcross, but it tells us there had to be an F1 also. What's more, this is a bird caught as an adult, which means it was born or it hatched and it flew south, wintered in India or Thailand or Bangladesh, and then flew back north again before we caught it. So it's a pretty healthy bird, really. It's lived a full year at least and has done pretty well. What's more is its F1 parent did pretty well, did the migration and actually had an offspring, right? And so this is telling us that these birds are not nearly as reproductively isolated as we might have thought. And one other note here, though, we only have direction of backcrossing in that direction. We haven't found any backcrossing in this direction. Notice the blue cluster, it's not like drawn out like the other one is. And when we look at detailed genotypes, so what I'm showing now is we have two chromosomes, chromosome 23, chromosome 26, and I'm showing just the high FST, so that means just high differentiation markers above FST of 0.95 between these populations. In each row is an individual bird. We've got the West Siberian ones up at the top in blue, East Siberian ones in red. And you see the West Siberian ones all have that dark purple sort of homozygous genotype at these loci. Most of the East Siberian birds have this light purple homozygous genotype in most of these loci. But there are some birds that have these long spans of heterozygosity. So I show those little triangles here. So whole strings across this divergent region of heterozygosity. And in the two chromosomes, there are different birds that have those heterozygous patterns. One bird in East Siberia even has a homozygous West Siberian chromosome on this chromosome, but a fully East Siberian chromosome on this side. So you see there's abundant evidence here, aside from our one backcross individual, of a trickle of gene flow from west to east across this meeting area of central Siberia. But you don't see any of the reverse. We see up in the blue population, we don't see any of these light purple things up there. So this is kind of telling us that there's this backcrossing only in one direction. And it's pretty logical to conclude here that hybrids must have lower fitness overall. I mean, they apparently cannot interbreed back with one of the populations. And even in the one direction they can backcross in, it's reasonable to think there's probably some lower fitness there. So we think that this potential to hybridize likely limits their range expansion into each other's range. And then a third example I wanted to put in really quickly, because Aidan, when he invited me, said that he was interested in three-way hybridization. And so the best case study that we've done of that is these Spheropicus sapsuckers done by Libby Natola, Dr. Libby Natola, a recent graduate from my lab, now a postdoc. And so we have the Spheropicus are one of these groups with a west coast form, an interior west form, and an eastern form. And they've always been called three species. So the red-breasted sapsucker, the red-naped sapsucker, and yellow-bellied, pictured here. An interesting thing is red-breasted and red-naped are more genetically related, but yellow-bellied and red-naped are more phenotypically similar. So there's something interesting has gone on there. But what Libby did was she went around and caught birds all over the place, took blood samples and sequenced them, and especially in this three-way contact zone. And this is just a map of her sampling in Western Canada, just all the different sort of phenotypes that she observed and their locations over there. This is showing an admixture plot. Just what I've shown here is just the kind of pure red-naped, red-breasted, or yellow-bellied individuals. Each individual is a column here. Now I'm going to show red-breasted and red-naped hybrids. So we've got a lot of different hybrid ancestry there. We've got red-breasted and yellow-bellied hybrids as well. And then we have yellow-bellied and red-naped hybrids. And more interestingly, though, for Aiden at least, and for many people, we have these tri-species hybrids, birds with ancestry of 3 different species being in the mix. We can look at these on a PCA. So up in the upper left, we have principal components analysis, again, showing what the birds looked like with their symbols and colors, and then genetically where they fall on that. So the good news is most of the time, phenotypically, that predicts genetically what they'll be, with some exceptions. But what you kind of notice is there's relatively few birds in the middle of that plot. So we can look at that another way with this triangle plot, where we actually plot red-breasted ancestry, yellow-breasted bellied ancestry, and red-naped in this tri-hybrid zone. And we find birds that are, primarily on the edges of this, but relatively few in the middle. And when we look at mating patterns, when Libby looked at mating patterns, these lines are between female-male pairs that were mated. So we're seeing there's a fair amount of assortative mating when they choose mates. So we now think, because of this assortative mating and the lack of many individuals right in the middle, that there must be low fitness of hybrids overall. So we think this is a 3-species tension zone where low hybrid fitness and some assortative mating prevents complete blending or range overlap. So we can kind of come back to these main explanations for lack of sympatry that I talked about. And I think it's pretty clear these examples that we're uncovering are examples where the hybridization itself, even if extremely rare, is likely really limiting range expansion and coexistence. And that's consistent with the modeling results as well. And we still have a lot of work to do to kind of integrate and ask in these systems, what role is ecology playing? Is that also, are these cases where they're so ecologically similar that they can't really coexist? Is that part of it? Or is it that they're ecologically different? We don't really know yet. So we're expanding the HSAM models to explore ecological variation, how that comes into it, and other spatial factors. And we're doing this now in 2 dimensions. And so ultimately, what I'd like to do is figure out a way to link up the simulations with real world cases. And so these dots I'm putting aren't exact. This is more of a conceptual, like where we'd like to be able to go. We'd like to be able to measure things from real populations and sort of put them on these maps of the simulation results. But it's actually rather hard to think of how do you measure things from empirical populations to match these parameters in the models. Because the parameters in the models you can define mathematically, but it's a little hard to know how to measure that empirically out in the field. So some take-home messages, hybridization or reproductive interference is likely to be a major influence on many species range boundaries. We need to better integrate coexistence theory and Klein theory. Simulations can provide much insight regarding range limits, hybridization, speciation, et cetera. I know that's not a message to convince many of you. Andy and his lab and many of the rest of you are doing simulations and working on the mathematical theory to answer a lot of these questions that I'm bringing up, so that's great. To learn more about the simulations that we've done, there are 2 papers here that you can look at or contact me. And this is just a bunch of key collaborators. on the work that I showed and a lot of funding to acknowledge. So thank you very much. I appreciate your attention. Don't know if we have time for questions or.
Oh, yeah.
Yeah, okay. I love questions.